Okay, this is the continuation of the sterilization and disinfection. I think the last thing that we spoke about was iodine. Just to cover, what are benefits of using iodine in dentistry. Um, these few points are very important. One reference shows that it has been shown to reduce spontaneous bleeding after tooth extraction, significant reduction of swelling after removal. Here, this is a third molar removal. Reduced oral caries in susceptible children. So حكينا مهم يعني susceptible children or children who usually have dysbiosis of the oral cavity. يعني they have growth of certain microorganisms over others, and they have an imbalance in their oral cavity, and that makes them more susceptible to caries and gingivitis. So if you want to reduce this susceptibility, you want to recover the balance, and certain mouthwash حكيناهم, and here using this PVPI can help reduce this in susceptible children. Effective antiseptic for these cones that are used in practice. And it does not discolor the teeth or irritate the mucosa زي ما حكينا بالمحاضرة الماضية with chlorhexidine based therapies. هلا بدنا ندخل نحكي عن ال heavy metals. So these heavy metals هلا يعني رح تشوفوا إنه there's a common theme that is coming in the following slides, which is we are still covering the protein effect. We are destabilizing the proteins and enzymes of the bacteria. So these are things that are already present. منهم heavy metals. So mercury, silver, these have the greatest antibacterial activity of the heavy metals. إذا سمعتوا عن مثلا using copper as for surfaces مثلا صحون, ملاعق, كاسات. These surfaces that are copper based وجدوا إنه they inhibit bacterial growth usually by destabilization of the membrane. So heavy metals, the presence of heavy metals, ولما تذكروا كنا نحكي بالجينيتكس عن إنه these horizontal gene elements مثلا plasmids ينتقلوا بين بعض البكتيريا، مش بس الانتي ميكروبيالز، كمان to be able to resist مثلا certain environmental stressors ومنهم ال heavy metals مثلا. So they act to disrupt proteins and enzymes by binding to the self-hydro groups in these enzymes. في thimerosal and mibromin both contain mercury. These are common metals used as skin antiseptics فقط على السكين. في شيء كتير مهم اللي هو silver nitrate drops. This is something else that we can give to babies that are born when the mother have an active STD مثلا gonococcal inflammation or infection. They are born and their eyes are exposed to the pathogen. So this is one of the things that can be used to prevent gonococcal ophthalmia neonatal, which is the condition. And these eye drops are given to these neonates at birth. راح نحكي كمان راح نقطة ثالثة راح يكون في antibiotic that we can use also for the same thing. And this can treat or prevent this condition. Without treatment, obviously, this can cause blindness. Silver, sulfadiazine, is also used to prevent infection of burn wounds. So this is also something that is based on heavy metals. This is the condition that where you can use nowadays. احنا بنستخدم antibiotic اللي هو erythromycin, but heavy metal based eye drops were used previously. اللي عمكم حدا فيكم شاف هذا these kind of tubs that are used. These have antimicrobial properties. Silver sulfadiazine, which is a self-drug antibiotic bound to silver, and it is an inhibitor of enzymes that is essential for folic acid synthesis. زي ما هم بيشتغلوا سلفا دراجز رح نحكي عنهم. The combination مع بعض اللي هو السيلفر مع السلفا دراج has a very broad spectrum activity, and binding to cell components مثل ال DNA can be responsible for this inhibitory effect. هلا كانوا يستخدموا هذول for partial and full thickness burns, but it is being phased out for other forms of topical antiseptics. Hydrogen peroxide. نفس الهيدروجين peroxide, there is the oxygen species that is used to destroy DNA. نفسه هو you can use it itself as an antiseptic to clean wounds. So this is a good antiseptic, and sometimes it is used to disinfect contact lenses rather than just keep them in normal saline wash. It is affected, is limited حكينا زي ما حكينا is by the organism's ability to produce اللي هو catalase, the enzyme. So as a catalase, it can degrade hydrogen peroxide. So the more that you put, the more that you can overcome the enzyme. طبعا هذا كله is يعني up to a point where toxicity might happen. هلا عادة مثلا بنعمل hydrogen peroxide activity catalase. انت بتعمل برجوكم بالمختبر how we do the catalase test. بتحط البكتيريا نفسها على catalase أو عفوا على hydrogen peroxide, it will start to bubble. That means there is catalase and it is catalyzing the reaction. So it is producing the breakdown products of hydrogen peroxide. So it is an oxidizing agent that attacks sulfhydryl groups, thereby inhibiting the enzymatic activity. شو بنستخدمه؟ هلا these can be used for sterilization of surfaces. Can be used to sterilize surgical equipment and tools, and can be used in vapor form to sterilize entire rooms. It has broad spectrum activity against viruses and bacteria, and obviously this works usually more on the DNA, and bacteria that harbors the enzyme obviously catalase will show resistance. حكينا يعني if you give it more time or concentration هي المعادلة اللي حكيناها, you can circumvent this issue. Environmentally it's safer than using bleach, but although it can bleach the skin. How does it bleach the skin? It can bleach بيستخدموه على الشعر كنوع من تبييض الشعر, يعني بيزيح من لون الشعر, وكمان على skin the same thing. So if you use it, it can bleach the skin as well. So they are using بيستخدموا مثلا أنواع من الكريمات للتبييض بيكون it has actually some portion, some of them have a portion of hydrogen peroxide that is it's bleaching basically the skin. تمام. خلصنا هاي النقطة. هلا بنحكي عن formaldehyde and glutaraldehyde. Both are available usually as يعني مخففين بمية. We don't use them neat. So 37% solution عادة is what is being used, and this can denature both proteins and nucleic acids. These formaldehydes and glutaraldehydes are used to preserve مثلا proteins, or used to preserve corpses or something that are used for عادة anatomy lab, but they can also be used as a form of sterilization technique. Glutaraldehyde has two reactive aldehyde groups and is 10 times more effective than formaldehyde, and it is less toxic. بالمستشفيات عادة نستخدمهم to sterilize respiratory therapy equipment. endoscopes, hemodialysis equipment, because mechanical and electrical sensitive equipment عادة ما بتقدر تستخدمهم. So one of the mechanisms where we can sterilize these equipment تحطهم in formaldehyde or glutaraldehyde عادة, and that has a good bactericidal, sporicidal, and virucidal activity. So it can actually achieve sterilization in enough time and concentration. Ethylene oxide, this is more extensively used than glutaraldehyde. It is less, it's more controllable أكتر. And يعني المستشفيات عادة اللي عندهم more capacity, more funding, that usually use ethylene oxide as a method to sterilize heat sensitive materials, هم surgical instruments, plastics, حتى كمان mechanical and electrical equipment. How does ethylene oxide work? It is an alkylating agent, so it by alkylating both proteins and nucleic acid, again the same thing. And they have this hydroxyl group that attacks reactive hydrogen atoms on essential amino acids and hydroxyl groups in the nucleic acids as well. So بيخربوا DNA, بيخربوا proteins, so they work indiscriminately and they can achieve sterilization. So ETO, ethylene oxide sterilizer, الجهاز بيشبه عادة جهاز الأوتوكليف، يعني من برا بيشبهه نفس الإشي. There's a point here where you have يعني this door that achieves pressure, capsule pressure، يعني بسكر الجوا الاريا as to prevent gas escape، فبيعمل لك pressure جوا and then you release the gas, ethylene oxide gas. بما انه gas, it will permeate everywhere. You give it some time and it will sterilize your equipment. So there is, you can use some temperature, add to the temperature to the equation as well, and that can improve yield. هلا لقدام شوي بنحكي مثلا we know، هلا احنا حنتذكر حكينا salts, acids, pH, temperature are usually, these are things that can denature proteins وخابل proteins. So all of these are things that we can use as sterilization and antiseptic mechanisms. حتى جسمك نفسه مثلا بيستخدم acid بالstomach، بيستخدم temperature بالفيفر، ممكن يستخدم مثلا يعني maybe not usually salt, but these are mechanisms that even our own bodies use. So we can use them ourselves. So strong acids and strong alkalis both kill by denaturing protein, much like we said. هلا much like heat, if proteins aren't made to withstand this certain pH, they will be denatured. Most bacteria are susceptible. It is important إن نتذكر نحكي إنه microbacteria are resistant to مثلا relatively resistant to NaOH at 2%. مثلا هذا ونحن نستخدمه، مثلا ممكن نستخدم NaOH 2% to kill all other bacteria and to improve the yield of TB. مثلا أنا باخذ عينة من بني آدم بدور على TB، بحطها to selectively kill all others because I know Mycobacterium tuberculosis is resistant to 2% NaOH مثلا. هلا weak acids مثلا citric acid, benzoic acid, that can be usually used to preserve food. هلا as food preservatives عادة بيستخدموا مثلا إذا بتستخدم weak acid it can be bacteriostatic، بمنع إنه البكتيريا تتكاثر. ممكن نستخدم salts، بيستخدموا salts عادة. وإيش كمان بيستخدموا temperature، مثلا بخلوه in cold، مثلا بخلوه على 4 degrees or -20 or whatever. And all of these are mechanisms to preserve food, to maintain food bacteriostatic. None of them are bactericidal. So weak acids can be used in this mechanism. So the action of these acids partially function to the organic moiety as well as the low pH. هلا نحكي عن modification of DNA, nucleic acids. هلا the things that we talk now about is basically more geared towards modification of nucleic acids. A variety of dyes اللي هم استخدموا مثلا زي حكينا crystal violet can be used as a skin antiseptic because نذكر حكينا it's part of the McConkey agar as can kill gram-positives. وعلى skin, the majority of bacteria that can kill or that can cause infection is gram-positive مثل staph or strep. So this is something that can be used as an antiseptic. It's actually based on binding and to positively charged dye molecule to the negatively charged phosphate group اللي هي موجودة على 5' end of the molecule, the DNA. So مثلا malachite green اللي هو مستخدم is part of the leucine ginseng medium, which is used to grow tuberculosis. This is something that can kill these gram-positives. The dye inhibits the growth of unwanted organisms. Again, the same thing. I want to kill other organisms and I want to make sure that the organism I'm looking for survives. So I look for something that it is resistant to, while others are sensitive to. هلا more physical agents. Physical agents that are either by imparting energy in the form of heat, radiation, or removing organisms by filtration. Heat. Again عن الأسد. هلا بنحكي عن heat. Heat energy can be applied in 3 ways. You can either give moist heat. Moist heat عادة اللي هو steam. يعني مثلا so you have you add steam that can permeate. بتشوفوا مثلا في دعايات هالأيام تنزل مثلا إنه بستخدم some sort of mop إنه ينظف الأرض فيها، وهاي المop is bound to a water bottle or something that can instantaneously pressurize and boil the water into steam. So بصير تعمل release steam and بحكي لك it kills 99% whatever of bacteria on the floor, which is true. It is a very good mechanism. مثلا بستخدموها على الكنبايات or whatever. So that is something that has been used in the past. هلا هو المويست هيت ستيم هو اللي استخدم الاوتوكليفing actually. So that's what we're using in autoclaving. Steam itself is a very good mechanism to بجوز هلا هالايام due to the coronavirus they're mentioning how انه مثلا اذا if you inhale steam that can kill the virus if any viral growth in your مثلا upper respiratory tract in the nose and can prevent إذا مثلا if you have been exposed to someone. قاعدين ببيعوا أو بيعملوا services إذا شفتوها مثلا in certain Southeastern Asian countries أو in China إنه مثلا بجيبوا it's actually a little bit funny but بجيبوا مثلا tubes بركبوهم على حيط والتيوبس طلعوا steam وبيجوا الناس بيجوا بدفعوا مثلا whatever money and then they sit in front of the steam and they breathe the steam and chant prayers or chant مثلا some things or whatever. So it is actually all because of the moist heat effect on DNA and again what else we will have to say also protein. So هذا أول شيء. ثاني شيء dry heat بحطه بنار أي شيء تحطه بنار يعني it will يعني بترفع درجة حرارة basically it will increase the heat. The other thing is pasteurization هو نوع من أنواع using heat إنه تذكروا أو تعرفوا عن pasteurization. Basically it's bringing the temperature of milk to about 55 or 65 degree and then rapidly cooling it بسرعة. So that kills a lot of the bacteria that cause مثلا a lot of diseases للناس مثلا brucella or other dairy borne organisms. فكل هذا reduces a lot of bacteria or pathogens in milk. So heat kills by denaturing proteins, but membrane damage and enzymatic cleavage of DNA may also be involved. Moist heat sterilizes at lower temperature than dry heat, and the water aids in the disruption of the non-covalent bonds. So that's why we use steam rather than dry heat in autoclaving. And the moist heat sterilization usually by autoclaving is the most frequently used method of sterilization. هذه نقطة كتير مهمة because bacterial spores are resistant to boiling at 100 degrees but must be exposed to higher temperature. This cannot be achieved unless the pressure is increased. If you increase the pressure, you can achieve higher temperature and then especially for the water, and then this will kill the spores. Now this kills even the highly resistant spores of Clostridium. احنا عادة to test the effectiveness of autoclaving، مثلا عندك autoclave بدك تعرف هل هو sterilizer is it sterilizing or not. بتخبي احنا بنجيب مثلا spores of Clostridium بنحطهم على plate، بنحط هذا البليت بالاوتوكليف، نعمل we run an autoclave cycle، بنطلع على البليت and we try to grow، بنحط مثلا broth على الspores we see if they grow. If they grow معناته they weren't killed by the autoclave، معناته autoclave ما له مشكلة. If they are all dead معناته you have achieved sterilization. So that's how we test the effectiveness of the autoclave. Sterilization by dry heat or by other مثلا hand required temperatures مثلا 180، بدك توصل ل 180 لساعتين عشان تعمل هذا اللي تعمله بالاوتوكليف. So it is actually it saves you more energy to do autoclaving. حكينا عن pasteurization. So it's used primarily for milk, consists of heating to 62 degrees for 30 minutes بعدين rapid cooling flashed. Pasteurization هلا الأيام استخدموه for 7 to 15 seconds. This is sufficient to kill a lot of the milk-borne pathogens مثل brucella, listeria نحكي عنها هاي، salmonella, streptococcus كلهم نحكي عنهم. Mycobacteria bovis also is a common pathogen in milk. So it does not sterilize the milk, but احنا حكينا we removed all the pathogens but not all the organisms. هلا المilk actually has a lot of good bacteria that you need to digest your food, to break down products that are released by the liver and especially in the bile. So you need bacteria to have that. So you don't want sterile milk, you don't want sterile cheese, you don't want sterile yogurt, you want these organisms. فهلا صاروا ينتبهوا انه these organisms a lot of them die when I pasteurize the milk. فناس هذول يعني hippies or whatever, they say okay I'm not gonna pasteurize the milk, you're killing the milk or whatever. But يعني we can remove the pathogens. احنا لازم نستخدم العلم. We remove the pathogens then we re-add all these organisms. يعني عادي نطلعهم, we pasteurize the milk and then we re-add them. So that can be done. So pros and cons. هلا استخدام autoclave versus ethylene oxide. Both they can achieve sterilization, but ethylene oxide is effective. You use it at lower temperature 50 60. It is reliable and شوية سريع. It sterilizes most material excluding liquids, foods or drugs. You can sterilize plastic, paper, electronics. مشكلته انه may have long cycles, toxic byproducts. الغاز ما بتقدر تنفسه اذا if it leaks it is very toxic. غالي مش رخيص. بعد ما تخلص السايكل بدك تعطيه فترة طويلة عشان الإثيلين أوكسيد يروح. You are using a gas and requires special safety handling. هالأوتوكليف safe, effective, economical, reliable, fast. بنضيف لك stainless steel, Pyrex, selected rigid materials, a lot of things that you can use, but you cannot use obviously electronics or certain sensitive material. عادة ما بتقدر تستخدم اشياء مش كويسة انه مثلا you are using wet heat material, corrosion ممكن يعمل لك corrosion على اغراضك over time. Some heat sensitive material you cannot use autoclave for. هلا تذكروا حكينا بنعرف ان UV light can break the DNA as we mentioned. So we can use UV light. وعادة هذه الاشياء بيستخدموها المختبرات انه مثلا اذا انا I'm preparing DNA بخلي البنش تبعي عليه دائما UV light to break all other DNA بطفي ال UV light لما اجي اشتغل انا فيه عليه. Or I can use UV light to in a room مثلا مشغل UV light in a room اذا ما فيها حدا and it will still destroy the DNA of all living organisms جوا. So you can achieve a good amount of sterility or disinfection using UV light. الويف لينث عادة مستخدمة between 250 to 260 because this is the maximal absorption of purines and pyrimidines. And remember, this is the formation of thymine dimers that we are aiming to do. As a result, replication will be inhibited and a lot of mistakes. حكينا هذه. So كله حكيناه هنا. هلا because UV radiation can damage the cornea and the skin, the use of UV radiation medicine is very limited. Skin cancer is usually due to a type of UV light, so we can't use it as a مثلا antiseptic. So it can be used in hospital to kill airborne organisms مثلا زي ما حكينا in certain rooms. بشغل هالاوبريشن روم، خلصوا منها الجراحة ما فيها ناس، بشغلوا ضوء واليو في and then it will kill a lot of organisms inside this surgery room. Same thing like we mentioned on benches where you use the DNA. مختبرات مثلا اللي فيها تشخيص كتير، بتستخدم PCR أو بتستخدم microbes مثلا growing microbes. You can keep the مثلا lab overnight on UV light and it will kill a lot of things. هلا البكتيريا suppose راح يكونوا resistant. They are obviously quite resistant and require a much higher dose of UV light to cause any damage to DNA than vegetative bacteria. X-ray حكينا هاي من الأشياء كنا نحكي عنها in genetics and in the growth إنه they can damage the DNA. حكينا X-rays they have a higher energy and penetrating power than UV light. عادة مثلا UV light ما بتقدر تصور، ما بنستخدمه بالتصوير، بنستخدم الإكس-راي بالتصوير because it can penetrate the body. So that's why we use X-rays and can kill a lot of organisms by production of free radicals. حكينا how they work by how they bind the DNA. So not only they target the DNA but also proteins and membranes by causing oxidative stress to these structures. هلا self-hydrocontaining compounds can protect DNA from these free radical attacks. So some other mechanism that is proposed is it has a direct hit on the covalent bond in DNA, which can cause DNA chain breakage. So X-rays will kill vegetative cells readily, but spores are markedly resistant again because again spores are not actively replicating. ما عندهمش repair mechanisms، مش قاعدين بيعملوا replicating بغير DNA. ف any damage that you cause it is not repaired or not registered. So and then the penetration of these rays is harder in spores. So شو بنستخدم X-rays? You can use X-rays again much like you use UV light and مثلا for sterilization of heat sensitive material مثلا sutures, surgical gloves, plastic items and syringes. You can use X-rays to do this. هلا بما انه this is a mechanism that causes، فدير بالكم انتم عادة dentists have an X-ray machine بيستخدموها، بكون فوق الشير تبع البيشنت وهو بيشتغل بله يصور which is fine. He has to do it, but there is a good amount of abuse here. Again بدك تعرف انت بدك تحمي حالك، بدك تحمي مريضك، لان خاصة انت يعني انت بتشتغل and you are one of those dentists that use an X-ray machine overhead. بدك تدير بالك كل مرة بتشغلها you are exposing yourself to a good dose of radiation. So you have to be very careful when you use them. إن شاء الله بيعلموكم how to use them, how to be safe in using these machines. So be very careful using these machines. Filtration, this is the preferred method to sterilize solutions. عادة solutions will be heat sensitive. مثلا تحط فيهم certain chemicals or certain proteins or certain things that you don't want to إذا بتعرضهم لمثلا الهيت they will it will مثلا يعني remove the properties of this solution that you need. So ما بنقدر نسخنهم. عادة مثلا من زمان لما كانوا يستخدموا solutions مثل intravenous solutions كانوا they will boil them وعشان to break down all bacteria inside and بروح بيعطوا السوليوشن للناس as an IV solution. فشو بصير هلا انت البكتيريا كانت موجوده هناك، تكسرت البكتيريا، واحنا حكينا انه الجرام نيجاتيف سيل وول خاصه حكينا has مثلا endotoxins within the cell wall itself. So as you break the lipopolysaccharide will be released, and lipopolysaccharide حكينا بيعمل هو septic shock, right? It can cause fever. So صاروا شوفوا الناس بكل ما نعطي اي في واحنا سخنا ويبول دي اي في الناس قاعدين بصير معهم حرارة. This is because of all the dead bacteria that is inside these IV solutions. فعشان هيك بلشوا نعمل filtration instead. عادة actually in the lab filtration is now a very common method used to sterilize small مثلا small amounts of solutions. مثلا أنا يعني كنا نشتغل in our labs in the UK and the US. عادة مثلا عندك you want to use something that's 1 mil, 5 mil or something 10 mil. We have these small filters, and then you inject them through the filter and you effectively sterilize the solution. You can use it right away بدل ما توديه تعمل له autoclave أو مثلا something else. So these are things that can actually used. شو ال basic mechanism تبع filtration؟ فلترة. شو الفلترة؟ فلترة you use a specific size pore. يكون عندك مثلا إشي some surface that has pores of a specific size. عادة pore size of 0.22 micrometer will filter everything. هذا السيرفس المستخدمه هو عادة is a nitrocellulose. nitrocellulose at 0.22 micrometer will filter out all bacteria, filter out all viruses, filter out all their byproducts, all the proteins, everything, and will let only your solution through. الأشياء المستخدمة مثلا نستخدمها كثير بيستخدم فيها مثلا vaccines, IV solutions. These are things that you are going to give the patient inject into the patient. عادة you will have to, or use on the patient, in the patient you will use filtration for them. فهلا test yourself on this table. What do you use to disinfect a surgeon's hand prior to surgery? So if you remember, we said this is surgical scrub, مثلا chlorhexidine. Disinfect surgical site prior to surgery. What do you use? So surgical site prior to surgery. So that means skin prior to surgery. So this is a major operation. وتذكر حكينا iodine. So بدك تعمل venipuncture or immunization. So we can use tincture of iodine. تذكروا حكينا مثلا or alcohol. بدك تعمل disinfect skin prior to blood culture, something larger than a venipuncture. مش alcohol. راح نستخدم هون tincture of iodine. You want to cleanse wounds. You can use شو تستخدم مثلا for wounds. If you think about it, you can use حكينا مثلا hydrogen peroxide, chlorhexidine. Burn wounds حكينا sulfur diazine. This is something that can be used for that. Clean up of blood from a patient مع hepatitis B or C, whatever. عندك area نزل فيها blood, whatever, patient has anything. So على أرض, what do you use? The best thing that you can use whatever you want, the highest thing. You can use مثلا هون مثلا bleach. Sterilized surgical equipment, and they are heat sensitive. مثلا شو تستخدم ethylene oxide or glutaraldehyde حكينا. Non-heat sensitive material مثلا gowns, drapes, whatever, you can use an autoclave. Intravenous solutions حكينا filtration. Disinfect air in an operating room. ما في شي ناس مش موجودين مثلا حكينا ultraviolet light. This is something that you use. Floors you can use مثلا QACs or whatever. Stethoscope قبل ما تستخدم مثلا ethanol. You can use preservative in vaccines, مثلا thimerosal, something that can be used as well. Food حكينا شو مثلا نستخدم weak acids. These are things that you can use, but these are all clinical uses. Halogen halogen releasing agents. These are the most important types of chlorine releasing agents. sodium hypochlorite, chlorine dioxide مثلا. These can be also work as an oxidizing agents. disrupt proteins. At high concentration they can be sporicidal. So again, the table for spores is very important. The feature of the spores, how they are resistant to heating. We can use here an autoclave, high resistant to many chemicals, so including most disinfectants, and you have to use methane solutions as sporicidals to kill the spores. They can survive for many years, especially in the soil. So wood contaminated by soil can be infected by these spores, مثلا tetani, perfringens, clostridium, these clostridia. نحكي عنهم بحالات gram positive rods. They exhibit no measurable metabolic activity. معناته antibiotics are ineffective against them. Spores form when nutrients are insufficient, but when they germinate when there is nutrients. So that's why they are not found at the site of infection, because nutrients are not limiting here. Spores are produced by members of only these two genera اللي هم Bacillus species and Clostridia. So نحكي عنهم إن شاء الله بوقتها. So again, proper sterilization of your surgical equipment, regular cleaning using gauze and saline, disinfection, sterilization of your equipment. This improves your outcome دائما مع your patient. This improves your methods دائما. This prolongs the life of your equipment. كتير أشياء يعني بتحكي عن أهمية ال proper use and the disciplined use of these methods at all times. So it is very important to maintain. This is a very important way of life for healthcare workers اللي بيشتغلوا دائما مع المرضى. So it is very important to maintain this. لما تستخدم مثلا IV، لما تستخدم venipuncture، بدك تستخدم مع المريض، بدك تعطيه إبرة مثلا to block a nerve. You are using your equipment to مثلا this remove saliva or to drill into the teeth. Whatever you are using, you have to make sure you're always following these sterilization techniques and these antiseptic methods. يعطيكم العافية. I think this is the end of this lecture. المحاضرات القادمة antimicrobials are very important. 3 محاضرات. بعد ما نخلصهم this will be the we'll have a small talk about the beginning of clinical microbiology, bacteriology. لهون بنكون نحن نخلص الميد، يعني محاضرات الميد. بعد هيك رح نبلش نحكي عن gram positives gram negatives، بكون هذول المحاضرات الفاينل. لهون يعطيكم العافية. We will begin with next lecture زي ما حكينا antimicrobials.